Welcome back to MedStudious. Today, we are taking a microscopic journey into one of the most vital, yet often overlooked support structures in the human body, reticular connective tissue. Have you ever wondered how incredibly soft, squishy organs like the liver, the spleen, or your lymph nodes maintain their distinct shape without collapsing into a puddle of cells? The answer lies in an intricate microscopic web called the reticular network. This tissue serves as the definitive architectural scaffolding for the body's soft organs. Think of it as an internal biological skeleton made not of bone, but of delicate branching threads. This entire network is constructed primarily from a specialized protein known as type 3 collagen, which forms the reticular fibers. In this video, we will deconstruct this biological blueprint, examine the specialized cells that build it, explore how it directs immune function, and look at crucial clinical applications, like how this exact tissue helps doctors stage cancer. Let's get started.
To truly understand reticular tissue, you have to understand the fundamental concept of the stroma. In histology and anatomy, we divide an organ into two distinct parts, the parenchyma and the stroma. The parenchyma consists of the workers, the specialized cells performing the actual function of the organ, like lymphocytes fighting a disease. But these workers need a building to work inside. That building is the stroma. The stroma is the supporting framework holding the workers in place, composed of connective tissue, blood vessels, and nerves. Let's use a visual analogy. Imagine a massive skyscraper. The parenchyma are all the office workers inside, doing the daily tasks. The stroma is the load-bearing steel skeleton and concrete foundation holding the entire structure up. If you remove the steel skeleton, the building collapses. In the body's softest organs, reticular connective tissue is that steel skeleton, forming the core mesh network that maintains the organ's physical integrity.
When we deconstruct this structural tissue, we find that it is built upon three essential pillars. Layer 1 is the cellular component. This is the workforce of the tissue, consisting primarily of resident reticular cells that act as the master builders, alongside a reserve of multipotent mesenchymal stem cells ready to repair any damage. Layer 2 is the fibrous component. This is the structural steel we just talked about. It is a dense, interconnected, mesh-like network forged entirely from specialized type 3 collagen fibers, known simply as reticulin. Finally, layer 3 is the ground substance. If the fibers are the steel, the ground substance is the biological mortar. It is a clear, amorphous, gel-like substance that fills all the empty space between the cells and the fibers, allowing nutrients and oxygen to diffuse through the tissue. Together, the ground substance and the fibers combine to create the complete extracellular matrix.
Let's zoom in closely on the fibers themselves, because their engineering is entirely unique. A reticular fiber is not just a naked string of protein. At its core, it contains type 3 collagen arranged in distinct structural fibrils. Surrounding this core is a middle sheath made of amorphous material. But the most fascinating part is the outer sheath. The resident reticular cells actually extend their own cell membranes to completely wrap around and encapsulate the collagen fiber. Because these fibers are hidden inside this cellular wrapping, they are practically invisible when you look at them under a microscope using standard H&E stains. This brings us to an important diagnostic note for medical students. To actually see reticular fibers, pathologists must use specialized histological techniques involving silver compounds. When stained with silver, these argyrophilic fibers turn a stark, visible black, allowing us to map the hidden network.
Now, what surrounds these silver-stained fibers? The ground substance. In reticular connective tissue, the resident cells are not packed tightly together like bricks in a wall. There is a significant physical distance between them, and this gap is bridged entirely by the ground substance. From a composition standpoint, this substance is a clear, highly amorphous, protein-rich gel.
Its primary function is to act as a biological shock absorber. It physically supports and suspends both the cellular workforce and the fibrous reticulin network, maintaining the precise 3D spacing required for the organ to function. If you were to point to a slide of reticular tissue, the identity of the ground substance is essentially everything in the tissue matrix that isn't a visible cell or a distinct structural fiber. It provides the hydrated environment that allows immune cells to swim through the network to hunt for pathogens.
Let's meet the specific cells that live in this environment. The first and most important are the reticular cells. Morphologically, they have a single nucleus and function very similarly to a standard fibroblast. You will find them interspersed throughout the tissue, heavily clustered at the junction points where the reticular fibers cross. Their function is critical. They are the primary manufacturers. They secrete the type 3 collagen and then extend their membranes to wrap the fibers, regulating how other cells interact with the network. Sharing this space are mesenchymal stem cells. These are the ultimate biological source code. They are multipotent stem cells found throughout all connective tissues, including as a major component of embryonic mesenchyme. Their primary job in an adult is to serve as the replacement engine. If a reticular cell dies or is damaged by infection, these stem cells can differentiate into brand new reticular cells to seamlessly repair the network.
So, where exactly does the body deploy this highly specialized scaffolding? If we look at an anatomical distribution map, we see it heavily concentrated in solid organs associated with filtration. Reticular tissue forms the entire structural scaffolding of your spleen, your liver, and every single lymph node in your lymphatic system. Furthermore, it is incredibly abundant deep inside your bones, forming the primary framework of your bone marrow, particularly within the axial skeleton of adults. It also has a role in microarchitecture, acting as insulating wraps. It forms delicate microscopic support networks that wrap around individual fat cells, muscle cells, and even Schwann cells, which insulate your delicate nerve fibers. Finally, as we just mentioned, it is a broadly distributed structural precursor in the form of embryonic mesenchyme, laying the groundwork throughout the developing fetus long before adult organs fully form.
By deploying this tissue in these specific locations, the body facilitates 3 core operations. Operation 1 is providing structural scaffolding. It acts exactly like a skeletal system for soft tissues, providing a static, unmoving physical framework that holds the organ's highly active functional cells firmly in place so they can do their jobs. Operation 2 is establishing cellular transit systems. The reticular fiber network isn't just a wall, it functions as a physical walkway or highway. During an active immune response, it physically guides B and T lymphocytes, as well as antigen-presenting cells, to highly specific destinations within a lymph node. Operation 3 is embryonic organogenesis. Before a complex, functional organ like a liver can grow, it requires a pre-existing 3-dimensional framework to grow onto. Reticular tissue builds this initial structural blueprint in the embryo, allowing the organ to take its final shape.
To really grasp how this structure enables function, let's look at a mechanism in action, the spleen's filtration system. The spleen filters your blood, and it relies entirely on reticular fibers to do it. Phase 1 is anchorage. Deep within the red pulp of the spleen, large immune cells called macrophages physically attach themselves to the reticular fibers that span across the splenic blood sinuses. Phase 2 is stationary sampling. Because they are physically tethered to the steel skeleton, these macrophages can remain perfectly stationary, resisting the continuous rushing flow of lymphatic fluid and blood. Phase 3 is assessment and destruction. As millions of red blood cells rapidly flow past, the anchored macrophages sample them. If they detect an old, faulty, or misshapen red blood cell, they grab it, pull it from circulation, and completely break it down. Without the reticular fibers to hold onto, the macrophages would just wash away.
When we look at an activated lymph node, We see that reticular cells actually specialize based on their exact location. A lymph node is essentially divided into different zones. In the deep zone, also known as the T-cell cortex, we find a specific subspecialty called fibroblastic reticular cells. This is their primary operational domain, where they interact heavily with T lymphocytes and the high endothelial venules that bring cells into the node. However, if you move outward to the superficial zone, specifically the subcapsular sinus just beneath the outer capsule, the environment changes. This is the operational domain of marginal reticular cells. These highly specialized cells extend their reticular networks deeply into the B-cell lymphoid follicles, creating a distinct microenvironment. By dividing the label, the lymph node can simultaneously manage both T-cell and B-cell immune responses on separate, highly organized structural networks.
Let's break down the functional divergence between these two subspecialties. The fibroblastic reticular cells act as the facilitators. Their job is support. They physically anchor massive numbers of macrophages and lymphocytes perfectly in place. They also manage transit, providing the continuous physical walkways to guide the movement of these immune cells. Finally, they handle filtration, manufacturing the actual molecular sieve that physically traps unwanted viral or bacterial particles. Conversely, the marginal reticular cells act as the messengers and architects. Embryonically, they construct the foundational framework required for the initial organogenesis of the lymph node itself. In the adult, their transit role is highly specific. They forge specialized fibers that ferry dendritic cells directly from the outer sinus straight into the B-cell follicles. This direct transit pathway is what triggers the targeted production of antibodies to fight off an infection.
As future medical professionals, you must understand how this tissue behaves in pathology. Our first clinical application is cancer staging and the sentinel node. Step 1 is invasion. A rogue cancer cell breaks away from a primary tumor mass and enters a lymphatic vessel. It flows through the lymph fluid until Step 2, entrapment. The migrating cancer cell hits the very first lymph node in the chain, known as the sentinel node. Remember that molecular sieve? The reticular fiber mesh reliably and inadvertently physically traps the cancer cell, preventing it from immediately traveling further. Step 3 is proliferation, where the cell establishes a metastatic stronghold inside the node. Because this reticular trap is so reliable, surgeons will locate and biopsy this specific sentinel node. By analyzing if cancer cells are trapped in its reticular web, pathologists can precisely stage how far a patient's cancer has physically progressed.
Our second clinical application is tracing malignancies to their origins. Recognizing the exact histological difference between normal stroma architecture and abnormal malignant growth is vital for identifying aggressive tumors. While cancer usually originates in epithelial tissue, the cells of the connective tissue can also mutate. Diagnostic target 1 is reticulum cell carcinoma. This is a rare, aggressive tumor type that arises directly from mutated resident reticular cells, the very cells that build the network. Because of their location, this cancer usually originates deep within the bone marrow or inside the lymph nodes themselves. Diagnostic target 2 involves sarcomas. Sarcomas are terrifying cancers that arise specifically from cells of mesenchymal origin. If those multipotent stem cells, the ones responsible for generating all connective tissue, sustain DNA damage, they can proliferate wildly, creating massive, deeply embedded tumors that are incredibly difficult to treat.
Our final clinical application explores how we differentiate lymphoma via biomarkers. The clinical challenge is common. A patient presents with a swollen lymph node. Is it just actively fighting a severe infection, or does it contain a deadly lymphoma? The diagnostic key lies in identifying follicular dendritic cells. These are highly specialized mesenchymal cells that provide structural support specifically inside the lymph node's germinal centers. Pathologists will specifically test a biopsy to confirm their structural presence or absence to definitively diagnose the lymphoma. How do they do it? The mechanism relies on unique receptors. These follicular dendritic cells uniquely express CD21 and CD23 receptors on their surface. By applying targeted fluorescent antibodies that bind strictly to CD21 and CD23, pathologists can visually illuminate these specific support cells under a microscope, allowing for a rapid, highly accurate, and definitive cancer diagnosis.
To wrap up, let's review the biological blueprint of reticular connective tissue. Its fundamental structural role is to function as the stroma, providing a non-binding architectural framework that supports functional tissues in soft organs like the spleen, liver, lymphatics, and bone marrow. The building blocks consist of the cells, the reticular builders and mesenchymal stem cells, the fibers, the silver-staining type 3 collagen, and the mortar, the gel-like ground substance. Together, they form a flexible, resilient extracellular matrix. The functional and clinical imperative is vast. Physiologically, it guides embryonic organogenesis, provides transit walkways for immune cells to hunt, and acts as the physical framework to filter the blood. Pathologically, its incredible filtering mechanism traps migrating cancer cells, allowing for accurate staging via the sentinel node, while mutations in its native cells can originate rare carcinomas and sarcomas. Thank you for studying with MedStudios. If this helped you, be sure to subscribe and keep studying hard.